Next topic, translation inhibitors. Translation inhibitors. There are some antibiotics or toxins which inhibit protein synthesis in prokaryotes and eukaryotes. Some examples are streptomycin, chloramphenicol, tetracyclines, erythromycin, fusidic acid, cyclohexamide, then pyromycin. Streptomycin mode of action. It binds with 30S subunit of bacterial ribosome and causes misreading of mRNA at relatively low concentrations. Tetracyclines, it binds to the 30S ribosomal subunit and interferes with aminoacyl tRNA binding. Tetracycline prevents binding of aminoacyl tRNA to A site. Chloramphenicol mode of action, it binds to the 50S ribosomal subunit and blocks peptide bond formation through inhibition of polypeptide transferase, but does not affect the cytosolic protein synthesis in eukaryotes. Erythromycin, it binds to the 50S ribosomal subunit and inhibits peptide chain elongation and translocation step. Fusidic acid, it binds to EFG elongation factor G and block translation. Puromycin, it is a secondary metabolite that blocks protein biosynthesis. It is a structural analog of the 3' end of aminoacyl tRNA but differs from tRNA at the aminoacyl amino acid is linked to the ribose sugar at 3' end via an amide bond rather than ester bond. So, in translation process, a site of ribosome is occupied by puromycin. Now, peptidyl transferase links the peptide of initial tRNA of P site to puromycin. Since the amide bond cannot be cleaved by the ribosome, no further peptidyl transfer takes place and complex fail of the ribosome. Puromycin terminates protein synthesis by acting as analog of the tRNA charged with an aromatic amino acid. Inhibition of translation by puromycin. Puromycin acid insoluble, peptidyl transferase acid insoluble. Location of amino acid tRNA and F-met tRNA can be determined by puromycin release assay. Only F-met tRNA enters partial P site on 30S subunit bound to mRNA. F-met AUG, F-met tRNA and met tRNA can be released by puromycin. Only AA tRNA enters a site on complete 70S ribosome. AA tRNA like met tRNA cannot be released by puromycin. MOA of chloramphenicol binds to 50S subunit of the ribosome, inhibit peptide bond formation. Chloramphenicol transpeptidation inhibited. Then general mechanism of protein synthesis inhibitors. Aminoglycosides block the initiation of translation and causes misreading of mRNA. Macrolides prevent the continuation of protein synthesis. Chloramphenicol prevents peptide bonds from being formed. Linkosamides prevent the continuation of protein synthesis. Oxazolidinones interfere with the initiation of protein synthesis. Then streptogramins, each interferes with a distinct step of protein synthesis. Tetracyclines block the attachment of tRNA to the ribosome. Neomycin is a broad-spectrum aminoglycoside antibiotic drug that is derived from the metabolic products of Streptomyces fradii. Neomycin is a complex comprised of 3 components, neomycin A, B, and C. Neomycin B, also known as framicetin, is the most active component of the complex, and neomycin C is the isomer of neomycin B, making these 2 stereoisomers the active components of neomycin 1,2. Neomycin A or neamine is a moiety that conjoins two molecules of neomycin B and C together. Neomycin is active against both gram-positive and gram-negative organisms and mediates its pharmacological action by binding to bacterial ribosomes and inhibiting protein synthesis, which is crucial for the survival of bacteria. Molecular biology. Neomycin resistance is conferred by either one of two aminoglycoside phosphotransferase genes. Neomycin binds to the 30th subunit of the ribosomes and inhibits translation of proteins from mRNA. Neomycin binds to the 30th subunit of the ribosome and inhibits translation of proteins from mRNA. So, mechanism of action. Like other aminoglycoside antibiotic drugs, neomycin inhibits bacterial ribosomes by binding to the 30th ribosomal subunit of susceptible bacteria and disrupting the translational machinery of bacterial protein synthesis. Bacterial translation is normally initiated by the mRNA binding to the 30th ribosomal subunit and subsequent binding with 50th subunit of elongation. Then, diphtheria toxin. Diphtheria is a disease resulting from bacterial infection by Corynebacterium diphtheriae. Diphtheria toxin is a monomeric protein with 535 amino acid residues responsible for disease-lethal effects. Diphtheria toxin specifically inactivates the eukaryotic elongation factor EEF2, thereby inhibiting eukaryotic protein synthesis. Diphtheria toxin catalyzes the ADP ribosylation of a diphtamide, a modified histidine residue of eukaryotic elongation factor EEF2, thereby inactivating it. So, inhibitor. Chloramphenicol inhibits peptidyl transferase on the prokaryotic subunit. Cyclohexamide inhibits peptidyl transferase on the eukaryotic large subunit. Erythromycin inhibits translocation by the prokaryotic large subunit. Fusidic acid inhibits elongation in prokaryotes by binding to EFG, the GDP, in a way that prevents its dissociation from the large subunit. Paromycin increases the ribosomal error rate. Then puromycin, an amino acid tRNA analog that causes premature chain termination in prokaryotes and eukaryotes. Streptomycin causes mRNA misreading and inhibits chain initiation in prokaryotes. Tetracycline inhibits the binding of amino acid tRNA to the prokaryotic small subunit. Diphtheria toxin catalytically inactivates EF2 by ADP ribosylation. Next topic, operons. So, introduction. The term gene regulation means that the level of gene expression can vary greatly under different conditions. The term gene regulation means that the level of gene expression can vary under different conditions. Genes that have constant levels of expression are termed constitutive, sometimes called housekeeping genes. The benefit of regulating genes is that encoding proteins will be produced only when required. So, transcriptional regulation. Most regulation of gene expression is at a transcriptional level. Rate of RNA synthesis increased or decreased. Transcriptional regulation involves actions of two types of regulatory proteins like repressors and activators. Repressors bind to DNA and inhibit transcription. Activators bind to DNA and increase transcription. Then negative control refers to the transcriptional regulation by repressor proteins. Positive control to regulation by activator proteins. Then transcriptional regulation. Small effector molecules affect transcription regulation, bind to regulatory proteins, not to DNA directly. Effector molecule may increase transcription. Inducers, for example, bind activators and cause activator to bind DNA, bind repressors and prevent repressor from binding DNA. Effector. Genes regulated by this way are inducible. Effector molecule may inhibit transcription. Co-repressors bind repressors and cause repression to both DNA. To bind DNA. Repressor to bind DNA. Inhibitors bind activators and prevent activator from binding DNA. Genes regulated in this way are repressible. The TRP operon. First one, the TRP operon. the TRP operon or TRP operon. TRP in the sense it's tryptophan, not TRP, TRP operon. Okay. The TRP operon is involved in the biosynthesis of the amino acid tryptophan. The genes TRPE, TRPD, TRPC, TRPB, and TRPA encode enzymes involved in tryptophan biosynthesis. The genes TRPR, TRPL are involved in regulation. TRPR encodes the TRP repressor protein, functions in repression. TRPL encodes a short peptide called the L leader or leaders peptide, functions in attenuation. When tryptophan is present, the TRP repressor binds the operator and RNA synthesis is blocked. Promoter, operator, TRPE, TRPD, TRPC, BA, A, B, C, D, E. In reverse. So, DNA polymerase, repressor, tryptophan. In the absence of tryptophan, the repressor dissociates from the operator and RNA synthesis proceeds. Promoter, operator, TRPE, TRPD, TRPC, TRPB, A, RNA polymerase in that direction. Organization of the TRP operon or TRP operon and regulation via the TRP repressor protein. Co-repressor binds to operator and blocks transcription. Repressor, co-repressor form active complex tryptophan. High tryptophan levels, repression occurs. Alternator sequence is there. Organization of the TRP operon and regulation via the TRP repressor protein. Another mechanism of regulation, more tryptophan levels, attenuation occurs. Attenuator sequence, transcription begins but stops at the attenuator sequence. TRP structural genes. Organization of the TRP operon, regulation via the TRP repressor protein. RNA pole can bind to the promoter. TRP operon attenuator sequence. RNA polymerase transcription inactive TRP repressor cannot bind to the operator site. Low tryptophan levels, transcription of the entire TRP operon occurs. Attenuation occurs in bacteria because of the coupling of transcription and translation. During attenuation, transcription usually begins, but it is terminated before the entire mRNA is made. A segment of DNA termed as attenuator is important in facilitating this termination. In the case of TRP operon, transcription terminates shortly past the TRPL region. This attenuation inhibits the further production of tryptophan. The segment of TRP operon immediately downstream from the operator site plays a critical role in attenuation. The first gene in the TRP operon is TRPL. This encodes a short peptide termed the leader peptide. Region 2 is complementary to regions 1 and 3. Region 3 is complementary to regions 2 and 4. Therefore, several stem loop structures are possible. The 3-4 stem loop is followed by a sequence of uracils. It acts as an intrinsic role-dependent terminator. These two codons provide a way to sense if there is sufficient tryptophan for translation. Beginning of TRPL coding sequence. Sequence of the TRPL mRNA produced during attenuation. Therefore, the formation of the 3-4 stem loop causes DNA pole to terminate transcription at the end of TRPLG. Conditions that favor the formation of the 3-4 stem loop rely on the translation of the TRPL mRNA. There are three possible scenarios: high level of tryptophan, medium level of tryptophan, high tryptophan tRNA, low levels of tryptophan, low tryptophan tRNA. Domain 1, domain 2, domain 3, domain 4 TRP leader region. Repression occurs. Co-repressor. Organization of the TRP operon, regulation via the TRP repressor protein. Then co-repressor form active complex. High tryptophan levels, repression occurs. Co-repressor binds the operator and blocks transcription. Then tryptophan. Attenuation occurs. Sufficient amount of tRNA Translation of the TRPL mRNA progresses until stop codon. RNA polymerase pauses. Region 2 cannot base pair with any other region. 3-4 stem loop forms. Transcription terminates. Translation termination codon. Red. Tryptophan levels 3/4 forms. TRPL, TRPE, DNA. Possible stem loop structures formed from TRPL mRNA under different conditions of translation. Transcription occurs. So low tryptophan levels 2/3 forms. Leader peptide TRP codons, ribosome stalls. Region 1 is blocked. 3 to 4 stem loop does not form. RNA pole transcribes rest of operons mRNA for TRPE. Inducible versus repressible regulation. The study of many operons revealed a general trend concerning inducible versus repressible regulation. Operons induced in catabolism, like breakdown of a substance, are typically inducible. The substance to be broken down or a related compound acts as an inducer. Operons involved in anabolism, biosynthesis of a substance, are typically repressible. The inhibitor or co-repressor is the small molecule that is the product of the operon. Arabinose operon. The three structural genes ARAB, ARA A, ARA B encode 4 enzymes needed for the metabolism of the sugar arabinose in bacterial cells. ARAB, ARA, I'm going to pronounce this as ARA. ARA B, arabinose in the sense. ARA B, ARA A, and ARA D encode for the enzymes kinase, isomerase, and epimerase. Kinase, isomerase, and epimerase. Kinase, isomerase, and epimerase. Isomerase converts arabinose to ribulose. Kinase converts ribulose to ribulose 5-phosphate. The arabinose operon also contains the arabinose C gene, which produces ARA-C protein. The ARA-C gene regulates the expression of the structural genes and the ARA-C protein. Thus, the ARA-C gene is autoregulated. The presence of both arabinose and the ARA-C gene product activates the expression of the bad genes. Structural elements of arabinose operon: promoter gene for arabinose protein, regulatory gene, operator gene, promoter gene, structural genes. The ARA-C gene is adjacent to the ARA operon. It has its own promoter, AC. It encodes regulatory protein ARA-C. ARA-C can bind to 3 different operator sites designated ARA-L, ARA-O1, and ARA-O2. ARA-C protein can act as either a negative or positive regulator of transcription, depending on whether or not arabinose is present. Arabinose, ribulose. Positive regulation. When arabinose is present, it binds to ARA-C protein and changes ARA-C conformation. An arabinose ARA-C dimer complex binds preferentially to ARA-L and not to ARA-O2, which causes opening of the loop. This allows mRNA pool to bind to PBAT. If glucose levels are low, CAP complex binds to PC and active the transcription. ARA-O2, ARA-C protein, arabinose binding domain, linker region, DNA binding domain, etc. can be seen. Operon inhibited in the absence of arabinose. Operon activated in the presence of arabinose. RNA polymerase. Loop broken. Negative regulation. When arabinose is absent, the ARC protein acts as a negative regulator. ARC acts as a dimer and causes the DNA loop. Looping brings the ARA-L and ARA-O2 sites in proximity to one another. One ARA-C monomer binds to ARA-L and second monomer binds to ARA-O2. Binding of the ARA-C prevents the RNA pool from binding to the PBAD promoter. Transcription not occur. Low arabinose and high glucose. Look at the structure from white. Next topic, eukaryotic gene expression. DNA packaging in eukaryotic chromosomes. DNA beads on a string model. Histones, nucleosomes, helix, supercoil, chromosome loops, and folds can be seen. DNA is packed tightly around histone proteins to form nucleosomes. DNA packing prevents RNA polymerase from accessing gene. DNA is packed tightly around the histone proteins to form nucleosomes. DNA packing prevents RNA polymerase from accessing genes. Regulation of gene expression in eukaryotes. Gene expression in eukaryotes starts at DNA level and ends with an enzyme catalyzing a particular chemical reaction or with structural metabolic protein. Signal molecules and unbundled signals and regulatory proteins affect gene expression in eukaryotes. In addition, genes can be chemically modified to enhance or inhibit readability. Gene expression in eukaryotes is controlled by a variety of mechanisms. This can be placed on of these 5 categories like DNA level, replicational, transcriptional, post-transcriptional, translational, and post-translational. So, levels of gene regulation in eukaryotes. Chromatin, control over chromatin modification, gene on DNA, primary transcript, mRNA protein. So, RNA processing control, sorry for the disturbance, transcriptional control, RNA processing control, RNA transport control, and translational control. So after that, gene regulation at the DNA level. Genes are altered by gene amplification, gene rearrangement, gene loss, methylation of DNA, chemical modification of DNA. Gene amplification. In order to produce large number of specific proteins, many copies of genes can be generated. This process is known as gene amplification. Genes are amplified in some species at certain stages of normal development. So gene amplified, gene A, gene B. Gene amplified, many copies of this gene A, gene B, like that. Gene rearrangements. Segment of DNA may move from one location to another in the genome, associating with each other in various ways, so their different proteins are produced. For example, gene arrangement occurs in the B cells that produce antibodies, immunoglobulin. The heavy chain gene is generated by rearranging specific sequences for large precursor chromosomal sequence into a single exon that encodes the variable region of the heavy chain. V1, V2, V3, etc. Recombination. They combine in numerous ways, their combination. Constant region, gene rearrangements in immunoglobulins. Then gene loss. If genes are deleted or partially deleted from cells, functional proteins cannot be produced. For example, gene loss occurs during the development of red blood cells. Immature erythroblasts contain nuclei that produce mRNA for the globin chains of hemoglobin. As cells mature, the nuclei are extruded so that the fully mature red blood cell is devoid of nucleus and therefore no genes, and so it can no longer produce mRNA. Then gene loss. Genes with 4 different functions: duplication, divergence, subfunctionalization, neofunctionalization, then degeneration or gene loss. Then transposons or jumping genes. Transposons are short segments of DNA which has the remarkable capacity to move from one location to another or in the same chromosomes or so to different chromosomes. This process is called transposition. Transposition is frequent in non-essential genes and is highly regulated. They confer some advantage on host like antibiotic resistance development due to transposons. They are called molecular parasites that exist in symbiosis with the host. Gene silencing, the process by which gene is switched off by various epigenetic mechanism by RNA interference, RNA by ncRNA. Gene switching, the process by which one gene is switched off while a closely related gene takes up its function. Eugenics means above genetics. Reversible heritable chemical modifications of DNA or histone or non-histone proteins that does not alter DNA sequence itself. Then epigenome, constellation of covalent modification of DNA and histones that impact chromatin structure and modification of gene expression. It is epigenetics. Reversible heritable chemical modifications of DNA or histone or non-histone proteins that does not alter DNA sequence itself. Epigenome, constellation of covalent modification of DNA and histones that impact chromatin structure and modulation of gene expression. Epigenetic modifications, DNA methylation, post-translational modification of histones, like that. So, methylation of DNA, chemical modification of DNA. Methylation of eukaryotic DNA may play a role in the maintenance of inactive heterochromatin, non-transcriptional chromosomes. In eukaryotes, the cytosine on both strands may be methylated to induce 5-methyl cytosine. Most methylated DNA will remain inactive during differentiation and during the repeated cell divisions of that cell line. Methylation probably is most important in preventing the transcription of the genes intended to be permanently turned off. So, methylation of eukaryotic DNA may play a role in the maintenance of inactive heterochromatin, non-transcriptional chromosomes. In eukaryotes, the cytosine on both strands may be methylated to produce 5-methyl cytosine. Most methylated DNA will remain inactive during differentiation and during the repeated cell divisions of that cell line. Methylation probably is most important in preventing the transcription of the genes intended to be permanently turned off. Special enzyme methylase during DNA replication methylase methylate the daughter DNA strands. Some developmental abnormalities are related to mutation that prevent methylation. Removing methyl groups from nucleotides has been shown to activate a gene. The Barr body is an example of chromosome that is highly methylated. Globin genes are more extensively methylated in non-embryoid erythroid cells than in cells in which these genes are expressed. Then chromatin remodeling. Chromatin remodeling refers to the changes occurring in DNA that enable it to undergo transcription, such as DNA is called active DNA or euchromatin. Chromatin remodeling refers to the changes occurring in DNA that enable it to undergo transcription, such as DNA is called active DNA or euchromatin. The DNA that does not accessible to transcription is called inactive and is called heterochromatin. Acetylation and deacetylation are the main events in chromatin remodeling. When transcription is not required, acetylation of nucleosomes is reduced by histone deacetylase enzyme. Histone modification, acetylation is more in euchromatin. Active DNA contains binding sites for regulatory proteins. Euchromatin DNA is hypomethylated at cytosine residue of CpG sequences. Then DNA regulatory proteins. Proteins that regulate the gene expression, such as repressor, inducer, enhancer, silencers, need to bind to DNA with high affinity to the specific regions of DNA. These proteins show several motifs which are involved in this DNA-protein interaction. Helix turn helix motif, zinc finger motif, leucine zipper motif. Regulation at transcription. Role of promoters, enhancer elements, repressor elements, tissue-specific expression of enhancer and repressor, regulation by alternative RNA splicing, alternative transcription start site in the RNA, alternate polyadenylation sites, regulation by RNA editing, regulation to mRNA stability, regulation at the translational level. Chromosome is there, nucleus, euchromatin, heterochromatin, silent, euchromatin, active region, nucleosome, in histone complex, heterochromatin. This is a representation to show heterochromatin. Chemical modifications of DNA or histones that change chromatin structure. They affect non-covalent interactions between and within nucleosomes. Euchromatin, active, heterochromatin, silent. Scientists know of at least 16 different modifications of histones and 4 modifications of DNA. Then examples of chromatin modifications: gene, histone tail, histone methyl or methyl group, DNA inaccessible, gene inactive. Methylation of DNA and histones cause nucleosomes to pack tightly together. Transcription factors cannot bind to the DNA and genes are not expressed. So histone acetylation results in loose packing of nucleosomes. Transcription factors can bind to the DNA and genes are expressed. Histone tail, DNA accessible, gene active. Packaging of DNA into chromatin. not just condenses the DNA but also can regulate gene expression. Chromatin remodeling can initiate or repress transcription. Histone modifications, especially of the nucleosome core particle, influence chromatin formation and also influence gene expression. Chromatin remodelers are molecules of about 5 different families that modify and mobilize the nucleosomes by various mechanisms that are ATP dependent. Chromatin remodelers can slide nucleosomes, evict nucleosomes, disassemble nucleosomes, replace canonical histones with histone variants. Chromatin remodeling. Nucleosome barrier. Transcription will not initiate or will not continue after initiation. Loosen the tight wrapping of the DNA around the nucleosome to make the DNA more accessible to transcription machinery. Then chromatin remodeling. Histone modifying enzymes. Phosphorylation, acetylation or deacetylation. Ubiquitination, modifications that make the chromatin rigid and compact or open and less condensed. The histone tails are hotspots for modifying enzymes. Depending on the histone code, the nucleosomes can recruit different molecular complexes like molecular chaperones, chromatin remodelers, etc. Then chromatin remodeling. Deacetylation erases these codes, thereby leading to less transcription. The acetylation of specific lysines can form codes that can be bound to by several transcription factors, molecular chaperones, etc. Then chromatin remodeling. Chromatin remodelers, highly conserved ATPase subunit that disrupt histones ATP contacts. Conserved multi-subunit complexes classified into 4 distinct families. Nucleosome assembly, nucleosome sliding, nucleosome eviction, unwrapping, then dimer replacement and dimer ejection. Then chromatin remodeling. Chromatin remodelers are of 4 families: actin binding to acetylated lysines or histones, highly conserved domain of ATPase, gene amplification, increase the number of genes available for transcription, resistance to methotrexate by cancer cells. So gene amplification increase the number of genes available for transcription, and resistance to methotrexate by cancer cells. Cancer cells increase the number of genes for DHFR drug resistance. Then gene rearrangements occurs during immunoglobulin synthesis. IgG light chain mRNAs are coded by different segments or domains. Variable VL 300 coding sequences joining 5 constant 10. Difficult for immune cells to achieve immunological specificity. Then immunoglobulin gene rearrangements. We learned about this earlier. Germline configuration, D to J recombination, V to DJ recombination, transcription splicing, translation assembly. Transcription splicing, translation assembly. Then immunoglobulin gene rearrangements. Single VJC coding sequences recombine, generate single contiguous transcription unit, RNAP transcribe into single mRNA. Then alternative RNA processing. Primary transcript is processed differently in different tissues, achieved by alternative transcription start site, alternative polyadenylation site, alternative splicing and processing, alternative RNA splicing. Most eukaryotic genes possess introns, non-coding regions that interrupt the sequence that specifies the amino acids of a polypeptide. Each intron must be removed from the RNA transcript of a DNA in order for the coding sequence to be expressed properly. So genes with multiple introns present a curious problem to the DNA splicing machinery. These introns can be removed separately or in combination depending on how the splicing machinery interacts with the RNA. If 2 successive introns are removed together, the exon between them will also be removed. Thus, the splicing machinery has the opportunity to modify the coding sequence of an RNA by deleting some of its exons. This phenomenon of splicing an RNA transcript in different ways is apparently a way of economizing genetic information. Instead of duplicating genes or pieces of genes, the alternate splicing of transcripts makes it possible for a single gene to encode different polypeptides. Alternative RNA splicing. One example of alternate splicing occurs during the expression of gene for troponin T, a protein found in the skeletal muscles of vertebrates. The size of this protein ranges from about 150 to 250 amino acids. In the rat, the tryptophan T gene is more than 16 kb long and contains 18 different exons. Transcripts of this gene are spliced in different ways to create a large array of mRNAs. When these are translated, many different troponin T polypeptides are produced. All these polypeptides share amino acids from exons 1, 3, 9, 15 and 18. However, the regions encoded by exons 4, 8 may be present or absent depending on the splicing pattern, and apparently in any combination additional variation is provided by the presence or absence of regions encoded by exon 16, 17 and if 60 present, 70 is not, and vice versa. So alternative splicing of transcription from the rat troponin T gene, only 3 of the possible 64 different mRNAs are shown. Then galactose operon, prokaryotic operon. Now we are going to once again learn that galactose operon is a prokaryotic operon, codes enzyme of an amphibolic pathway of D-galactose metabolism. Galactose operon performs catabolic and anabolic function. Catabolic function, galactose is used as C carbon source. Catabolic function, UDP galactose, UDP glucose, galactose is used as a carbon source. Anabolic function, UDP glucose, UDP galactose. This leads to formation of lipopolysaccharide, cell wall component. Then structure, operator 2, operator external, operator internal given, promoter 2 overlapping, transcription start site, another one. Look at the figure. Then HBS, histone unit binding site at plus 6.5, activator sequence, structural genes, galactose epimerase, phosphate, uridylyl transferase, galactokinase, mutarotace, etc. Structure of galactose operon, GalR, CCC, operator AS region, then like this, ETKM. This operon, when performs catabolism, galactose imported into cell by permease or galactose formed intracellularly by disaccharide metabolism. In galactose metabolism, beta D galactose is not utilized. Thus, it's converted into alpha beta galactose. So, lactose, D-glucose, beta galactosidase, beta D galactose, alpha D galactose, galactose 1-phosphate, UDP galactose cell wall component, UDP glucose 1-phosphate. Mutarotace is the enzyme that links galactose metabolism into common pathway. GalR encodes repressor for gal operon. It is unlinked gene. Regulation of gal operon 4 types. GalR mediated DNA loop formation or repression. GalR gene encode, tetrameric repressor that binds to 2 operators. One at plus 55 and one at minus 60 site ON and OE. This process requires presence of histones like proteins and supercoiled DNA with operators in some force. Interaction of 2 operator bound gal and leads to formation of DNA loop repressome. This loop blocks RNA polymerase to sit on promoter, and this loop inhibits formation of open complex. Structure of genes are not transcribed. Condition 1 inhibits promoter P1 transcription of, inhibits formation of open promoter complex through RNA polymerase contact with alpha CDT, also causes contact inhibition of P1. Condition 2 activates promoter P2. This allows basal genes transcription of genes transcription on. Note the GAL-G1 complex will not cause any repression because of presence of antipose sequence in intermediate upstream area. Next condition, contact activation will be happening. 3. Induction of galactose operon by D-galactose. Condition 1. Increased level of galactose, both alpha and beta in medium. Galactose binds as allosteric site of dimer repressor. Conformational change in repressor. Suppress interaction of repressor with RNA, P and DNA. Induce operon and increased rate of galactose metabolism. Condition 2. DNA looping. D-galactose first breaks galactose R and galactose R tetramers into individual galacto-R-OE and galactose R-O1 dimer. P1 is repressed and P2 is activated. Gal-R-O1 dissociated by O-galactose. Gal-R-OE dissociated with P1 is activated. Then regulation of CRP-CAMP complex. CRP-CAMP complex binds at activation site 35 region. Blocks DNA polymerase to form open complex with P2. Transcription stops. CRP-CAMP leads to formation of closed complex at P1 with RNA polymerase that further isomerize to open complex. Transcription on. But when glucose concentration is increased in medium, CRP-CAMP concentration is decreased. Basal level transcription occurs at P2 by formation of binary complex. CAMP and its receptor protein CRP complex enhances P1 but represses P2. Next topic is about ribosomes. The ribosomes are small, dense, rounded and granular particles of the ribonucleoprotein composed of mainly rRNA proteins. They occur either freely in the matrix of mitochondria, chloroplast and cytoplasm or remain attached with the membranes of the endoplasmic reticulum and nucleus. Ribosomes exist as monosomes as well as polysomes. Monosomes are inactive solitary ribosomes, whereas polysomes are active functional group. Type 70S ribosome are comparatively smaller size, occur in the prokaryotic cells of the blue-green algae and bacteria. 77S and 60S ribosomes, the fungi generally possess 77S type, whereas animal mitochondrial ribosomes are 60S. 70S ribosomes occur in eukaryotic cells of the plant and animals. Then structure. The ribosomes are spheroid structures of 150 to 250 AO in diameter. Each ribosome is porous, hydrated and composed of 2 subunits. One ribosomal subunit is large in size and has a dome-like shape, while the other ribosomal subunit is smaller in size and occurring above the larger subunit and forming a cup-like structure. The 70S ribosomes consist of 2 subunits, 50S and 30S. The 50S ribosomal subunit is larger in size and has the size of 160 AO to 180 AO. The 30S ribosomal subunit is smaller in size and occurs above the 5OS subunit like a cap. The 80S ribosome also consists of 2 subunits, 60S and 40S. The 60S ribosomal subunit